synapse is a region where communication occurs between two neurons or between a neuron and an effector cell, such as a muscle or a glandular cell. The term presynaptic neuron refers to the nerve cell that carries a nerve impulse or action potential toward a synapse. It's the cell that sends the signal. A postsynaptic cell is the cell that receives the signal. So it may be a nerve cell called a postsynaptic neuron that carries a nerve impulse away from the synapse, or it may be an effector cell that responds to the impulse at the synapse, such as a muscle cell that contracts. The synaptic cleft is the area between those two cells. So the presynaptic cell has an area between it and the postsynaptic cell, and that area is known as the synaptic cleft. So it's the area between the two. So here we see a neuron, and we see several different types of synapses associated with this one neuron. So, of course, this is the cell body. Here are the dendrites, and here's the axon. So here we have the axon of another neuron coming in and synapsing with the axon of this one. So this is an axo-axonic synapse, which simply is a fancy term to say two axons forming a synapse. Here you have axodendritic, so the axon of this blue nerve cell is forming synapses with the dendrites of the yellow neuron, and axosomatic, the axon of the green neuron, is forming synapses with the soma, or the body, of the yellow nerve cell. So a synapse, remember, is the junction between the nerve cells and another nerve, a neuron, or an effector. So because we have these different types of cells doing different things, we classify them based on their structure and their function. So the structural classifications, we just saw some of those, such as the axodendritic, the axo-axonic, and the axosomatic. So those obviously were neuron to neuron junctions or synapses where they meet. Neuro to glandular, where the nerve cell is forming a synapse with glandular cells, and neuromuscular, such as we saw with muscle contraction and the motor neuron. Functionally, we classify synapses as electrical or chemical based on what they do. An electrical synapse has action potentials or action impulses that conduct directly between the cell membranes of the adjacent neurons through structures called gap junctions. So as ions flow from one cell to the next, the action potential spreads from cell to cell. These gap junctions of these electrical synapses are very common in smooth muscle of the viscera, the organs, the cardiac muscle, and also in the developing embryos. They also occur very frequently in the brain. Electrical synapses have two main advantages. They allow for faster communication because the action potential is conducted directly through those gap junctions. So electrical synapses are faster than chemical synapses. And also synchronization. So electrical synapses can synchronize and coordinate the activity of a group of neurons or a group of muscle cells. Chemical synapses are much slower in the way that they act. They're separated by a synaptic cleft, so the postsynaptic and the presynaptic cells don't touch. They're just close together, separated by that cleft. So in response to a nerve impulse, the presynaptic neuron releases a neurotransmitter that then is going to diffuse through the fluid in the synaptic cleft and bind to receptors on the postsynaptic cell. These chemical synapses relay signals more slowly than electrical synapses. In this image, we'll look at how signals are transmitted at a chemical synapse. So here we have a small picture showing what we're seeing here. So an action impulse or an action potential has traveled down this axon to the axon termini, and the little synaptic bulb, the enlargement at the end of the axon, and here we have the postsynaptic cell. So we have the dendrites of this cell. So here's cell number 1, cell number 1, cell number 2, cell number 2. So as the action potential propagates down the axon of this nerve cell and it reaches the synaptic end bulb or synaptic terminal or terminal bulb, you'll hear a lot of terms to describe this, that change in the voltage of the membrane causes calcium channels to open. As the calcium channels open, the positively charged calcium enters the area of the synaptic bulb, and in this synaptic bulb there are vesicles that are storing a neurotransmitter such as acetylcholine that we saw with muscle cell contraction. So the calcium comes in, it triggers these vesicles to fuse with the presynaptic membrane and to dump their neurotransmitter into the synaptic cleft. The neurotransmitter then diffuses across the synaptic cleft and binds to receptors on the postsynaptic cell. When the neurotransmitter binds to these receptors, it triggers these receptors to open, so they are ligand-gated channels. When that gate opens, it's going to allow ions to flow into the cell. Now, depending on what those ions are, that can either inhibit or excite that postsynaptic cell. So excitatory stimulation in the synapse can lead to sodium gates opening and causing the postsynaptic cell to depolarize. So that's what we call an excitatory synapse, where the neurotransmitter binds to the receptor on the postsynaptic cell and opens those sodium gates, which leads that cell, the postsynaptic cell, to become closer and closer to depolarization. So we call that an excitatory postsynaptic potential. An inhibitory synapse is one where the neurotransmitter causes hyperpolarization of the postsynaptic cell. So it is an inhibitory postsynaptic potential, and most of the time it does so by opening those potassium gates or potassium channels, which allow potassium to leak out of the postsynaptic cell, and that is going to hyperpolarize that membrane and prevent that cell from depolarizing. This is just a simplistic look at the two terms we just talked about, the excitatory versus inhibitory postsynaptic potentials. So the neurotransmitter here was released into the synaptic cleft. When it binds to receptors on the postsynaptic cell, it causes sodium to come into the postsynaptic cell. The cell depolarizes and an action potential is fired. On the other hand, an inhibitory postsynaptic potential hyperpolarizes the postsynaptic cell. So the neurotransmitter is released into the synaptic cleft. It opens ligand-gated channels that in turn cause potassium to leave the cell. Therefore, the cell becomes less depolarized and is less likely to fire an action potential.